Hey guys, how you doing? So this is the third part of Chapter 9, and in this chapter we're going to be mainly looking at stem cells. It's going to hopefully be a very short video, and we're going to finish Chapter 9 in this video and then move on to the next chapter. So we're going to look at stem cells and how stem cells sort of work in organisms. So let's jump straight into it. Okay, so stem cells. Now, before we understand stem cells, we have to understand that even though all organisms, all the cells in an organism will have the same amount of DNA, not all of that DNA is used. So what happens is some segments of the DNA is used by certain types of cells. Now, those cells are what we call specialized cells. So these can be your nerve cells, your blood cells or your liver cells, but the word specialized means that they have adopted a role and they are now locked into that particular role. Yeah, so they've specialized in that particular area. So when a cell becomes specialized, it goes through a particular process called a differentiation. It differentiates into that cell that it's going to be in the future. Yeah, if you want to think about your body as being like society, then each individual, each adult is like a specialized cell, yeah, with a particular function. If it's a nerve cell, it performs functions relating to the nervous system. If it's a blood cell, it relates, performs functions relating to the cardiovascular system. Before a cell differentiates, it is what we call a stem cell. So a stem cell really is just an undifferentiated cell which has the potential to replicate and develop into many kinds of cells. So it is like a blank cell, if you will. So in the same way, a stem cell is like a person in our society who has not yet taken on a particular job or role. Yeah. So this is like you guys as high school students. You guys are like the stem cells of our society. And when then they differentiate, they become specialized into those particular cells that they are going to stay. Yeah. Now, there are different types of stem cells. So the one with the most potential is called a totipotent stem cell, and these can be any type of cells necessary. Yeah, the only type of totipotent stem cell in an organism is a fertilized egg. So that fertilized egg, that single cell, the zygote, can differentiate and form every other cell within an individual. Yeah, so once it moves beyond that, the cell is no longer a totipotent, but it is what we call a pluripotent stem cell, which can become most types of cells, but not all. So the majority of embryonic stem cells are what we call pluripotent. They have the ability to change into many, many different types of cells, but not all. Yeah. So embryos are the kind of the predominant kind of area where we get those pluripotent stem cells from, and hopefully by now you sort of understand why the idea of embryonic stem cells creates a lot of kind of debate around that area. Okay. The next level is called the multipotent stem cells. So these cells are able to give rise to have a specific function. Yeah, so they're specific usually to a particular type of cell. So for example, blood stem cells have the ability to become blood cells, and they're called multipotent. So you can sort of see the degree of potential sort of decreases as you go down. Totipotent being the one that can change into the most types of cells, and then pluripotent, and then multipotent. Yeah, decreasing their potential as you go. Okay. Now, in mammals, let's go with a few examples of what stem cells can do. So basal stem cells in the epidermis. The cell cycle happens at a very rapid rate just beneath the skin. Yeah. The reason for this is because your skin and my skin is often replaced in 48 days. Every skin cell only lasts about 48 days or so and needs to be replaced in that time frame, which means that stem cells need to grow and multiply at a rapid rate to keep up with the amount of skin cells that are actually being lost. Yeah. Humans tend to lose about 30 to 40,000 skin cells every hour. So you can see the amount of skin cells that need to be reproduced at roughly the same rate. Yeah. Here's a diagram. So beneath the skin layer that you and I are familiar with, the top layer here is the dead skin cells. These are ones that have already died, and they form the layer on the outside, and they're the ones that you tend to lose every day, right? And just below those ones is the actual living tissue, which is the basal stem cell. So how it works is that a stem cell has the potential to become a skin cell. This stem cell here, the basal stem cell beneath the epidermis, is a multipotent stem cell and can become a skin cell, but it hasn't yet. So what it does is it will differentiate and divide Now, it'll divide and form two copies of itself. One version will stay as the original copy, and the other one will then differentiate and turn into a skin cell. Yeah, and then that skin cell again would multiply into two, and then they will slowly form the skin cells that you have. So for each basal stem cell, you can get two skin cells forming. Okay. Another example. So other than your skin, your intestines. So stem cells in the gut lining regenerates every 4 to 5 days. Now, every time food passes through your intestines, it takes with it a few cells in your gut lining. So you are losing cells in your gut as you digest your food. These cells that are being lost need to be replaced, and the cells that do that replacing are called intestinal stem cells, or crypts. Okay, here's a diagram of what that looks like. This particular sample here shows the gut lining, and the outer layer there is where the food passes through, and the villi of the intestines are up there, and down below are the actual crypts, the basal stem cells that are forming new gut lining every time. And it works kind of like a conveyor belt. Yeah, the basal stem cell shown in the bottom here with a diamond will differentiate. It will form two copies of itself. One will become the differentiated gut lining cell, and then the other one stays as a stem cell, so it can keep repeating that process. And it goes 1 to 2, 2 to 4, 4 to 8, and so forth, until you get your intestinal cells, which are then lost. And so you can see it moves slowly towards the outside, and then the dead cells are picked off and removed in the process of digestion. Okay. The last example we've got is the hemopoietic stem cells, which is blood stem cells. These cells are found in the bone marrow. So it's interesting that blood is not produced in blood is produced in the bone marrow. And so the bone marrow contains the stem cells required to produce blood cells. So all of your different types of blood cells, your red blood cells, your white blood cells, platelets, they come from a blood stem cell. Yeah, and the blood stem cell will differentiate into those different types of cells in order for you to have the function that the blood has. Yeah, red blood cells carrying oxygen, white blood cells for immunity, and platelets for clotting. So hopefully that gives you guys a nice idea of how stem cells work. We will look at stem cells in more detail and looking at embryonic stem cells and the development of embryos and some of the kind of ethical issues behind that as well. But for now, that's just a bit of an overview. Okay. Now, the last thing I want to talk about is how the cell cycle affects plants and fungi. Up until now, we've only been looking at how cell cycles affect animals in terms of mitosis, but it also affects plants as well. So, for example, in plants, not every part of the plant is alive. Yeah, the majority of the plant is actually dead and forms really just the woody tissue or the structure of the plant that keeps it upright. However, the part that is growing forms what we call epicormic shoots. So here's an image of a tree that has just been burnt, and you can see from the side of the trunk new epicormic shoots coming out from the side. Yeah, so that particular area is the growing part of the plant, and it only forms one particular layer. In just beneath the bark is the living tissue. Okay, in fungi you get a slightly different thing again. So if you ever look at bread and you kind of see that kind of fluffy stuff on top of bread mold, that fluffy stuff is what we call the spores of the bread, and the spores are connected by a mycelium, which is a network of fibers beneath the spores, usually throughout your piece of bread, and that mycelium is the growing part of the fungus. Yeah, and so if you actually see a piece of bread with a few spots on it and stuff, you're seeing the spores. The rest of the bread is actually already infected with the mycelium, and the whole thing should really be thrown out. Yeah, so that's that. Okay, so hopefully you guys get a good understanding of the cell cycle. We're going to move into chapter 10 in the next lesson. So I'll catch you guys then. See ya.